Hello everyone. Today we'll be speaking about molecular translation for efficient R&D. My name is Siddharth Kotwal and I'm joined by my colleague Sofia. Quantifi is a AI-first digital engineering services company. We are Nvidia's service delivery partner of the year for Americas, and we very closely work with Nvidia product teams such as Omniverse, Nemo, Clara, Monai, and Nvidia professional services to close the gap between the Nvidia products and what the customers want. Today we'll, like I said, speak about molecular translation and frame the problem statement and how deep learning can accelerate and improve experimentation in this important area of drug discovery. Next, we'll go over some experimental results and speak a little bit about Nvidia DGX Foundry and its benefits to accelerating the cycles of experimentation when it comes to really heavy models that we leverage in NLP in computer vision. And then we zoom out and look at the applications of NLP in the biopharma lifecycle and how you can work with Quantifi to accelerate your biomedical NLP projects. With that, I'll hand it over to my colleague Sofia.
Thanks a lot, Sid. Hello, everyone, from me as well. I am Sofia Moschou, and I have a PhD in computational astrophysics. Pleasure to be talking to you all today about a very interesting case. Prior to joining Quantifi, I have spent 4 years in Harvard doing postdoctoral research, and at Quantifi over the last year and a half, I've joined and I am a senior machine learning engineer helping to put together client solutions, and I also am keeping busy on the applied research side of things. Today we will discuss our work on molecular translation, which is no other than a task of translating images of molecules into text. This use case illustrates the type of work that we do here at Quantifi and the steps that we take to make our clients' workflows more efficient. At Quantifi, we have a very strong healthcare and life sciences team, and we carry out several workloads across the entire field. For this use case, we used data from a Kaggle competition. This particular dataset that we got to work with contains about 10 million images of chemical substances and associated annotated text. The task is to annotate those images that exist in the test dataset and we don't have labels for by predicting some useful text sequence which describes essentially what's depicted within the molecule. In order to get some context on the applicability of this use case, it's important to think that while great technological developments are shaping the world we live in, sometimes the best and easiest tools are still graphical representations on pen and paper. Chemists, even today, often write down molecular formulas using the same notations as have been used for centuries. In more recently published research, molecular images have already been annotated with machine-readable chemical descriptions. However, there is still a plethora of scanned documents that can't be automatically searched for specific chemical depictions. And that's the exact need that molecular translation is coming to answer. Molecular translation is a sequence prediction task that can help us interpret old chemical images by converting them back to their underlying chemical structure, and essentially looking at an image and predicting an associated text sequence. In our case, this sequence is in the form of a formatted chemical notation string that is called InChI, or otherwise International Chemical Identifier. There are other standard chemical notation formats, such as SMILES, that can be considered, but in this case we will discuss about InChI. When this process of predicting the InChI sequence from molecular images is automated and applied to a large volume of scientific papers, it can help chemists expand their access to collective chemical research and identify novel trends by mining large data sets. Finally, and maybe the most important point of them all, is that with molecular translation, what we can do is speed up R&D efforts by identifying previously published molecules and excluding them from downstream tasks, for example, in drug discovery cycles. Next, we will discuss the overall architecture we put together for the sequence prediction task. If we exclude for the moment the pre-processing and the post-processing steps of the approach, at the very core of the workflow lies an encoder-decoder type of architecture. For this work, we were inspired by the Show, Attend and Tell paper that was written and published in 2015 by Zhu et al. On the lower half of this slide, we can see an intuitive example of this approach. Let's assume that we have an input image of a flying bird over a body of water. The encoder is essentially performing feature extraction and passing a feature map down to the decoder. The decoder in turn learns through an attention mechanism to generate words when it sees some associated features in the image. For example, it learns to specifically detect the bird, and it also learns to count different bird-looking structures. So, for example, in this instance, the first word that it predicts is A because it's counting a single bird. Then it identifies the body of the bird. So the second word is bird. It identifies that it's flying over water, and so on and so forth. It's predicting the entire sequence. Essentially, for each input image, we get some text sequence that describes the contents of this image. You might have heard of this task referred to as image captioning. In our case, the images depict specifically molecular structures, and the text is following some formatting rules that comply with the InChI notation, which we will talk about more later on. So we go from the image to this particularly formatted string. Next, let's take a closer look into these InChI sequences and the information that is encoded therein. In general, as shown on the top two panels of this slide, molecules can be pretty complex three-dimensional structures, consisting of many elements and having many different types of bonds and having also quite different and specific stereoscopic three-dimensional configurations in space. As we mentioned earlier, InChI stands for International Chemical Identifier, and it's a text sequence that describes chemical substances. The InChI notations were designed to provide a standard way to encode molecular information while maintaining the same level of structural details provided and the same conventions for illustration purposes. Every InChI sequence starts with a string like InChI equals, as can be seen at the bottom part of the slide, followed by 1S for the version number. The remaining information is structured in a sequence of layers and sub-layers, with each of them encoding some specific type of information about the molecule. Layers and sub-layers are separated by the / delimiter. The main layer is the one that's indicated by the dashed box, and it contains the most important amount of information. The main layer consists of 3 different types of sub-layers. The first one denotes the chemical formula and is shown with red letters here. The second one is talking about the atomic connections, and it's the carbon sub-layer. It's shown with yellow letters. And the last one is showing the atomic bond information of hydrogen. It's shown with green letters. In the chemical formula, is the classic notation that we learned at school, where we write one element after the other in a specific order, also mentioning their multiplicity and so on. The atom connection sublayers are showing for each of the carbons, for example, how many and what types of bonds we have, and the same goes for hydrogen. Finally, after the main layer, we have another 5 layers that provide some extra information on charge, stereochemical information, and isotopic characteristics of the molecule. So to sum up, InChI sequences contain detailed information about chemical substances that are human-readable after some training. For example, chemists can very easily, by looking at this particular InChI string, visualize in their mind what a particular molecule looks like, what its properties are, and so on. In order to get a better intuitive understanding of the information within an InChI string, it's important to note here that the chemical formula alone, the thing that we learned at school, is not enough of a discriminator. This is because many molecules with vastly different properties and illustrations share the same chemical formula, as can be seen in these 4 examples on this slide. What we can see in these examples is that we have many different molecules that share the same chemical formula, which I remind you is only one part of the InChI sequence. It's the main layer, one sublayer of the main layer. But at the same time, all these different molecules have quite different overall InChI sequences and very different visual representations, as we can see in this slide. So the InChI sequence carries a lot of information about the molecules beyond just their chemical formula, for example, how molecules are oriented and connected in space. As in almost all the use cases, we have to do some pre-processing on the original data. Here we have 2 different types of data: the molecular images and the associated InChI sequences. What we do is that we pre-process, we do some pre-processing in both image and InChI strings. For the images, we do a few standard pre-processing steps, including cropping and resizing them to a uniform image size for our images, as well as to invert them to a more computer-friendly representation of black and white colors. In that representation, the background is black and the molecular structures are white. For the InChI strings, the pre-processing is very similar to what you would do for a natural language processing problem, or for example, translating sentences from one language to another, for example, translating an English sentence to French. As shown in this animation, in this slide, the classic steps here are to split the text sentence into the words that it consists of. This means that we break down each string down to some subset of substrings. If we do this for all the sequences or all of our InChI strings in our case, and we only keep the unique words or tokens, we can construct a chemistry-specific language. This chemical language can then be used to construct all the InChI strings within our dataset. And this is what we did in this case. The other thing we need to do is to pad all our sentences or InChI strings, in our case, to the same sequence length. That length is determined by the longest instance in our dataset. Eventually, the last step that we do is to transform these string tokens to numbers. And this is because our machine learning downstream models that we're going to use as downstream tasks work better than they prefer, in a sense, to work with numbers rather than with text. More particular in our case, our sentences are formatted in this InChI format that we discussed in detail earlier. So our start string token is always the same, defined as the string InChI equals 1s, as shown in the first green bubble. There is no specific end token like the sign period that we put for normal human language. So at the end, we need to make up one end token. So what we do is exactly that. We add an end token at the end of each sentence. Now that we have the start and end tokens, what we can do is to tokenize the in-between parts of the sequence. That in-between text is the one that makes up all these layers and sub-layers that we discussed earlier on in our InChI string, and they are separated with this forward slash delimiter. Essentially, what we're doing is, through this process, to come up with a vocabulary or some sort of chemical language where the letter C represents carbon, and the letter H represents hydrogen, the number 10 represents multiplicity, and so on and so forth. Our chemical language, in this case, has 300 unique words, which are these symbols that I just described. Finally, all of our sentences or InChI sequences are padded to match the longest existing InChI sequence in the dataset, similar to what we did for the image side of preprocessing. We brought all of our images into the same size and so on. Next, we will take a closer look at this main part, which is the modeling part that we described earlier on as well. The first part we will discuss is the encoder part of the architecture. The encoder performs feature extraction on the preprocessed image. The preprocessed images can be used as inputs to the encoder that goes from full resolution image into a representation of lower dimensionality. The encoder achieves that by passing the molecular image through a number of convolutional layers and related layers, essentially downsampling it until a flat layer is reached. That flat layer holds the image embedding. For this work, we experimented with a variety of encoders from pretty much the complete suite of EfficientNet and EfficientNet V2 models of various sizes, and we also implemented a fully custom vision transformer as an encoder. So we have a variety of options here to use. I would like to emphasize already at this point that we're very proud to have put together a very modular architecture that allows us to replace the flavor of the encoder that we use in our module with an encoder of our choice, whatever that might be, and that might change in the future, without our architecture or pipeline changing significantly and with it still being able to work as intended. What this means is that whatever replacement of the encoder we do now or in the future, the pipeline will still get as input a molecular image, and it will generate an InChI sequence as a prediction at the end of the workflow. Next, we will take a closer look at the decoder. The decoder receives as input the flattened image embedding that was acquired at the previous step, together with the relevant InChI string, and using an attention mechanism, it learns to associate image features to specific parts of the InChI sequence. Essentially, the decoder is the one that provides the InChI sentence prediction for each image. Specifically, the decoder quickly learns that all InChIs start with the start token, then followed by the letter C that denotes the carbons, then followed by the multiplicity of the carbons. So it learns to search within the image through the attention mechanism and spot where the carbons are and count them. So this process continues, and we have the hydrogens, go and count the hydrogens until the end token is reached and all the features have been described within this particular molecular image. Similar to what we did for the encoder module, we experimented with a variety of decoders, ranging from stacked recurrent neural networks with GRU or LSTM units and attentions of type Bahdanau to a fully custom transformer decoder with self-attention. Needless to say that the transformer provides a huge speed up during training, as it does, essentially we can do a very efficient parallelization, and in our recurrent process, this is the one that is the most computationally costly, as you may already know. Again, I'd like to point out the importance of the solutions modularity, of this model's modularity, which allows us to reuse the developed end-to-end pipeline when we want to update some machine learning model with some better and newer, more favorable version in the future. This is important for both research and development R&D workloads, when speed of implementation and experimentation become very important, but also for operational efforts by shortening the life cycle of the development to deployment and operations. So that's the main idea for the main part of the architecture that we have already described that. Once we get our predictions from our decoder, we then perform a couple of post-processing steps. As the first post-processing step, and in order to boost our model's accuracy, we modify the method that's usually followed at the recurrent step of the decoder. Typically, during training, the decoder would get as input the feature map of the original image as computed by the encoder, together with the known at training InChI string label. Then recurrently, and at each step after the start token, the decoder would predict the next part of the sequence. For example, after the start token, we have a carbon, and after that, we have the number 10, and so on and so forth, until the entire InChI sequence is predicted. There are about 300 unique tokens, as I mentioned earlier, in our chemical language in this data set. So at each point, the decoder computes 300 probabilities for each of these tokens to be the next token in our sequence. Once the most probable step is computed, then we rank the probabilities of the predictions that we would typically, and what we would typically do is to choose always the top one scoring as our prediction of the next step in the sequence. This approach of computing all the probabilities and our prediction always being the top one, the most probable prediction for the next, is called greedy approach. That's the usual method followed. The method that we use instead, implemented and used instead here, is called beam search. So beam search differs with respect to the greedy approach that's usually followed, in that instead of at each step choosing the single top most probable token as our prediction, we keep the top 5 most probable scenarios and we keep them as viable options throughout the entire decoding process. You can think of the greedy approach as beam search with a single beam. We always have them keep the single most probable scenario. What beam search does by keeping the top 5 most probable scenarios as shown in this animation is that it boosts the model's performance by considering more scenarios and including information from future steps. This is demonstrated in the animation below, where initially the first beam is the single most probable one, but if we consider more scenarios or more beams and compute a few more steps down the line, then we see that the beam number 2 becomes more probable than the beam number 1. Now, without the beam search, without allowing more scenarios rather than the top one, then we would have missed this outcome, and the model accuracy would have been lower. In other words, beam search is allowing for more breadth and tractability when decoding. Please note here that implementing beam search is a post-processing step and it doesn't really affect our training. As the second post-processing step, we use an external Python library called rdkit. And this rdkit library is a Python library that is relating to chemistry. So essentially, it has it's a chemical library. More specifically, earlier on in this work, we realized that some of our predicted InChI strings, some of our predicted sequences point to non-existent molecules. So they are invalid InChIs. And we also realized that we can use this external rdkit library to get rid of, to eliminate all these non-existent InChI sequences and only keep valid InChI sequences that point to a particular molecule. Once we get rid of all the invalid InChI strings, then we are left with a set of only valid options, and we can re-rank them and submit our predictions with only valid options. We saw that that provided a significant improvement on our prediction step, on our prediction score. And this is the last step in our post-processing part of the workflow. Finally, in order to obtain our final predictions, we reverse the tokenization process and get the predicted InChI sequence for each input image. So we go from a sequence of numbers that were predicted by the decoder. We get rid of the padding, convert these numbers back to the associated string tokens, and finally bring it all together, and we obtain our final InChI text prediction, which is our final output of the workflow. The next thing I would like to talk about is the implementation challenges and the infrastructure that we chose for this work. In Quantified, we are an AI-first company, and we build custom engineering solutions for our specific client needs each time. As you can imagine, machine learning is a very important part of this process, but it's only one part of an efficient machine learning operations workflow. And other important parts to consider are the reusability and modularity of the code and the solutions we put together, the reliability, as well as the computational speed and cost. In order to build a successful solution, like what we did for this case, it all comes down to the way we address the challenges that are present in this particular use case. With respect to these three parameters that I mentioned before, reusability, reliability, and speed and cost. The challenges for this use case were the handling of big data, and I remind you that we worked with a dataset that included 10 million molecular images and associated InChI strings. Now, processing 10 million images requires that we make efficient use of the available memory, and we create a fast enough pipeline that can complete the computations as soon as possible, and as soon as possible in the order of minutes or in the orders of hours. For that, we put together a pure TensorFlow pipeline, making use of APIs such as the TF Data API, which makes it possible to handle large amounts of data, read from different data formats, and perform complex transformations. We use TF Data API to pre-process and transform our images and InChI sequences into TF records files. TF records files are a simple file format for storing a sequence of binary records. Transforming the original dataset into binary TF records has the advantage of using less memory and providing speed up for processing speed, and giving a speed up the processing speed for the ML models downstream. Furthermore, we use the TF function decorator that converts regular Python code to callable TensorFlow graph functions, which is usually more performant and can make better use of the hardware infrastructure that we are using. Other advanced optimizations that we use to boost our model's performance include mixed precision as well as accelerated linear algebra. So pretty much, to sum it all up, we used all that TensorFlow has to offer, which had a significant impact in the performance of this model and pipeline altogether. Now, right software engineering and hardware choices can have a huge impact on our workflow's applicability, on our models or our solutions applicability to tackle our clients' essential technical needs. Powerful hardware is what I'm going to talk about next. We recently tested this model and pipeline that I just described in detail on Nvidia's new GPU high-performance computing cluster that's called the GX Foundry. Next, I will discuss how this experience was for our team at Quantified. For completeness, before jumping into the takeaways from our own DGX Foundry experience, I'd like to start with a short introduction of Nvidia's DGX Foundry, bringing out and calling out especially the aspects that I found more important during this experience. DGX Foundry, for those that don't know about it, is a high-performance computing cluster that is powered by the next generation GPUs, and it can be used for accelerated machine learning and classic computational workloads. The Tensor Core GPUs of DGX Foundry are the A100 machines, and they can be clustered together. They can be used to run either single GPU or multi-GPU or multi-pod computational loads. Essentially, a pod or a node is a cluster of 8 A100 machines that work together efficiently. And we can use either a single GPU or multiple GPUs or entire multiple nodes or pods of these sets of 8 machines. Now, if you're working with large models, there is 640 gigabytes of GPU memory that are available to accommodate that, and the Foundry can reach 5 petaflops of computational performance. Now, as applied researchers and engineers in general, what we care about, one of the main things we care about, is the user-friendliness of a platform and the ability that it provides us to experiment quickly and do experiment cycles as fast as possible. I can already say that DGX Foundry was a great experience on that front, as we got the opportunity to utilize and familiarize ourselves for our workloads, all these machine learning operation integrations, and gain a lot of benefit out of interactive sessions that are possible in the Nvidia's platform. Getting early access and working on DGX Foundry was very beneficial for our team overall at Quantified. And the first thing, and that's for a number of reasons, the first thing I'd like to call out is the support and the interactivity that we got from the Nvidia team. And I'd like to say here that that was an exemplary support that we got. Anecdotally, I would like to mention that we even got support in debugging our code's parallelization to run more efficiently on a multi-GPU setup for our multi-GPU experiments. The command line and the graphic user interfaces were very user-friendly, and that allowed us to familiarize ourselves with the platform really quickly and set up in a very straightforward and reproducible manner our experiments and do parametric studies very easily without having to modify a lot of code each time. Another element that was of paramount importance to us was the comprehensive telemetry and also the logging functionalities that the platform allowed that made our lives much easier. With respect to profiling the code for its experiment, the Nvidia platform readily is providing useful information on the usage of GPUs, the usage of memory, the usage of network, and other very useful parameters as time series. So at all times, for all of your experiments, you will get a very nice visual representation of what percentage of GPUs is being used, what memory is used, and at what specific timestamps. On top of that, detailed logs of the code are also available, and you can have some control there on the types of outputs and logs that your code is keeping. And by combining these two elements, that is comprehensive telemetry and logs, what you can get is that you can very easily pinpoint any performance bottlenecks that your code may have, and you can then very easily go and debug and fix them if you know exactly where these bottlenecks occur and at what time and what specific part of the code this is an issue. In terms of performance, our code ran 5 times faster on an A100 GPU compared to Cloud T4 GPUs that we had tested at the same pipeline before. On DGX Foundry, we were able to train our molecular translation models on an entire pod or node of a set of 8 A100 machines, and we were able to reach a training performance and speed comparable to that we got from an 8-core TPU v3, which we had originally used to train this model and develop the model. Now, bringing all these elements together, the most important overall image that I'd like to paint here is that what DGX Foundry allowed our team to do is to shorten the research and experimentation cycles from an order of days down to the order of hours, and that is of paramount importance, and it's a fascinating prospect for the efficiency of our R&D and also business workloads. In conclusion, I would like to emphasize that we put together a powerful, modular, and very applicable solution. And the reason for that is its low cost and high speed. And we did this by combining software engineering innovation with powerful hardware architectures. The most important aspect here is that we applied these optimization techniques not only during training, but more importantly, also for the operational aspects of a model, is that we apply that in our inference pipelines. In order to give you a more intuitive explanation, I will put down some numbers to demonstrate the impact these two components, so software engineering innovation and also powerful hardware, can have in the efficiency of the final pipeline that we put together. So in order to do that, our model, I'm giving you some numbers on the time that the model takes to compute 250,000 inferences. So to provide InChI predictions, text sequence predictions for 250,000 input images. The Quantified model takes less than 15 minutes to perform inference on all of these 250,000 molecular images, while other state-of-the-art models that we found and we're presenting here took anywhere from 4 to 12 hours for the inference alone. To make this even more intuitive, let's associate some cost to it, assuming that we want to use this model for performing batch predictions after deploying it into production. So let's say that we want to host this model on the cloud and spin up an instance with some specific hardware when we want to perform those batch predictions. The associated cost for the Quantified model for each 250,000 images is less than $1, while at the same time, those other state-of-the-art models that we discussed before, and we anecdotally are mentioning here, were $24 or $50. And you might think, okay, that's not really that bad. But now, let's say that we are interested in using this model multiple times on a daily basis for a very large number of molecules, because it's important for some downstream, for example, drug discovery task, as oftentimes it's the case. So during drug discovery on the early stages, the number of molecules that we may be interested in playing around could reach multiple millions and so on, and we could try to replicate this pipeline multiple times a day. Then, if we scale this to a yearly cost for using this model, we can see that the Quantified solution comes down to being tens of times cheaper than these other solutions that I mentioned before. And the reason for that is that all these other state-of-the-art solutions may provide really highest accuracy, but what they did not take into account is that they were not fine-tuned for providing a sustainable and a practical operational solution for a healthcare and life science company. And oftentimes for a healthcare and life sciences company, what we care about is also to minimize operational cost. And that might be a very important aspect, and sometimes even more important than achieving the maximum accuracy. With that, I will pass it to my colleague Sid. Sid, can you tell us how does NLP, natural language processing, tie into the broader field of drug discovery, and how is Quantified working with Nvidia to engage with customers in the area?
Thanks, Sofia. I'll next talk about the application of AI, specifically NLP in the biopharma value chain. As you can see, the process of getting a drug to the market all the way from discovery to launch is fairly complex. It involves sourcing and supply of raw materials for production, manufacturing, distribution of the drug, and delivery to the end customer, and finally post-market surveillance. On an average, it takes about 10 years to bring a new drug to market. And as of 2014, according to Tufts Center for the Study of Drug Development, the cost of developing a new prescription drug that gains market approval is approximately $2.6 billion. So the returns on new drugs that finally get to the market do not favor massive investments that Biopharma makes in R&D. So let's discuss how NLP can optimize each step of this value chain. Take, for example, the first stage that is exclusively focused on research and discovery. At this stage, understanding gene-disease associations, biological pathways, and systems is extremely critical. Much of the data that can aid in extracting this information is in fact unstructured. Furthermore, most new targets are derived from new discoveries that push our understanding of the human body and are often published in scientific literature. So mining of such biomedical literature becomes very important for extraction of actionable information present in free text data. And NLP comes into play in the process by enabling automated techniques such as NER, relation extraction, that allow you to reach this end goal. NLP has also been utilized to aid various processes in drug discovery life cycles such as gene disease mapping, biomarker discovery, patient trial matching, adverse drug events detection, and these applications commonly use functionalities and large language models such as Biomegatron and BioBERT. Quantifi has currently been working with two leading biopharma companies in this domain to build a knowledge graph from external and internal data sources using such language models. Another application is drug repurposing, which is also a promising area for ensuring faster drug development and is focused on the discovery of new therapeutic opportunities for existing drugs. It can lead to fast approval process, safer treatment, and reduced healthcare costs. NLP is often used to extract drug-disease treatment pairs from EHR and clinical notes, which can be used in a downstream hypothesis as well. As we move to the next cycle, we look at clinical development and trials phase. Approximately 5.6% of clinical trials in the clinicaltrials.government database have been terminated prematurely as of 2021. And failure to optimize clinical trial design, inefficient enrollment processes, and poor retention rates are one of the main reasons for premature trial closure. Identification of suitable patients is extremely resource intensive and often involves manual review of clinical notes for identification of eligible patient, traversing complex hierarchies and codes where the information may be split between different systems. NLP techniques for automating clinical trial eligibility pre-screening for patients increases the efficiency of patient election and recruitment. Quantifi has developed an NLP-powered multi-channel virtual agent for Wake Forest Baptist Medical Center that allowed them to effectively assist several cancer patients to find clinical trials relevant to their conditions on the website. And finally, the tail end of this value chain is post-market surveillance. Active post-market surveillance is extremely crucial to understanding the side effects that can result from a drug in a larger population over the larger duration of its usage. Of course, this population will be much larger than the clinical trial, and hence monitoring adverse events is extremely important. But based on the several reports from regulatory authorities, adverse events are very, very rarely reported. In fact, they are very underreported. NLP can be used to improve the efficiency of this entire pharmacovigilance process, which involves performing various tasks such as analyzing EHR records, social media data, and conducting interviews. Remember that people are unlikely to go through the complex process of reporting issues and concerns and adverse effects to manufacturers or regulatory authorities, but are more likely to post on public forums or even Reddit. And biomedical NER models can be trained to identify medications from these posts, their attributes like dosage, frequency that led to this adverse drug event. And finally, understanding the interaction of two co-administered drugs can also become a very critical part of fully grasping drug adverse reactions, where again, NLP can be used to extract information from patient records and scientific literature. Now, oftentimes, customers that work with Quantify want to know if they have a problem statement such as building biomedical knowledge graphs for drug discovery. How do they go about it? How do they work with Quantify and Nvidia to ensure that this idea is built into a prototype into production? So this represents a canonical framework to build such applications. We call it hack it, prove it, nail it, and scale it. Hack it starts often with working with your line of business leaders and data scientists to engage in ideation and work on hands-on training sessions with Nvidia SDKs. It's a 2-day session that will allow you to understand the vocabulary of biomedical NLP, the state-of-the-art tools to train these models on GPUs, and then finally, how can you benchmark and what are the KPIs to look after. The next stage is called Prove It, and here we want to leverage the Nvidia NGC models and Nvidia SDKs to develop a focused proof of concept that addresses a specific need. This might be identifying certain gene names or certain drug therapies in your biomedical literature and training a named entity recognition model using Nvidia NeMo. Finally, when you do prove this out, you want to scale the initiative even more. You might want to develop a much larger biomedical knowledge graph, train language models from scratch, and that's what the nail it phase is focused on. Here, as we spoke earlier, Nvidia DGX Foundry or DGX SuperPods are some of the most active platforms that can enable you to scale your projects to production. And then finally, in order to truly inject and ensure that AI becomes the lifeblood of your organization's digital transformation journey, building a hybrid AI center of excellence becomes very, very crucial. And that's where Nvidia DGX infrastructure comes. And Quantify, together with Nvidia, can help you accelerate each step of this journey. And in order to learn more, you can reach out to us, book an ideation workshop that can help you explore how AI is transforming your industry, how can NLP help you leverage and accelerate some of your proof of concepts, strategize and prioritize certain use cases that address directly your business challenges, and then finally move towards building a pilot and an AI center of excellence. With that, I'd like to end today's session. Thanks a lot for joining us, and take care.
All right. Thank you so much, Sid, for wrapping up and giving us some more information about engagement models that Quantify offers. Thanks, everyone, for tuning in and listening to us. Now, let's jump into some questions. Please feel free to add any more questions you may have in the chat, and we will receive it on the back end over here and try to answer them one by one. All right. Let's jump into the first question. What was the evaluation metric that was used for this experiment? So the evaluation metric that was used for this experiment was the Levenshtein distance. Essentially, what we get as output is particular InChI strings that are these sequences, again, or in this special format. And the Levenshtein distance essentially is a metric that tells us how many letters in this entire string we got wrong. So in general, in 90% of the strings that we got were not longer than 200 or so characters. So for those, we got almost all of them pristinely correct, but with very long InChI strings, with very large molecules, we made some mistakes, and on average on the larger molecules, our mistakes were about 2 letters wrong, which isn't too bad. All right, next question. Why are transformers better than recurrent neural networks? So transformers, as we said earlier, they are better than recurrent neural networks, and the reason why they are able to achieve that is twofold. First of all, they are faster in processing the input than recurrent neural networks, as I remind you that vanilla recurrent neural networks need to wait and process the inputs one by one, pass it through all the layers and make the predictions. Whereas the transformer, because they have this very powerful trick that they do, which is called positional embedding, they essentially have the information of the entire sequence at once. And for that reason, they can parallelize the calculation, and they don't have to wait for the previous steps. And they can look both backwards and forward. And for that reason, because they have access at the entire string, at the entire sequence, for that reason, they also have higher accuracy than recurrent neural networks. So they're actually better because of these reasons. Another reason is also that oftentimes for not vanilla, but more GRUs or LSTMs, that they are more advanced recurrent neural networks, they do get some more information. They can be very complex, so they have many parameters, and typically the LSTMs are actually more complex than a transformer is. So complexity is the third reason that it's better than recurrent neural networks. All right, next question that I see. How does the vision transformer work, and is it actually better than CNNs? So this is actually a really good question. So how do vision transformers work? The vision transformers were the first part that we used. I remind you that this is a complex, actually a multimodal task that we do in molecular translation. We get in an image and we need to break it down and understand it and have a model that comprehends, does feature extraction and all that. So how the vision transformer works is that it gets this initial input image and it breaks it down into small patches. A typical, for example, size could be that's used is 16 by 16 pixels. So let's say we have a 1000 pixel by 1000 pixel image. So the vision transformer breaks it down into smaller patches, embeds all these different patches correctly, and then it also adds positional encodings on all these different patches. And with the self-attention mechanism, it learns to associate specific features at specific places of the larger image. The question whether it's better or not with respect to classic CNNs is a really good one. So it's a more advanced, it's a state-of-the-art model that seems to be quite robust. Essentially, if there are naturally corrupted patches, then it seems that the vision transformer is quite good at learning to ignore these and grabbing information from all the others intuitively, without me being really an expert on vision transformers. But the way I also understand it, that it works, is that it learns. So, for example, it learns through this attention mechanism to identify where in the image lies the most important information for the particular task that we are training on. So it learns to also ignore other parts that aren't very important for this particular task. So in that, it's more robust than CNNs. In general, maybe a downplay for vision transformers from a paper that was published last year, if I'm not mistaken, is that they require quite large data sets to achieve high accuracy. But if we have really many records, for example, many millions of images and examples, then the vision transformers can really outperform traditional CNNs for the reasons I mentioned earlier, and in terms of also computational cost as well, due to the positional encodings that they include as well, but also, of course, performance and accuracy. Any other questions you may have, please fill them into the chat space. Wait for a couple of minutes for that. All right, if there is no other question, thank you very much for your attention, for tuning in today. If there's anything else, if that you found that interesting, and if you would like to discuss further, please don't be shy and shoot us an email. We would be really happy to get back to you. Thank you all for joining us.